Thanks, Ed. Okay, so my job is to just review some highlights from the BioXFEL consortium of the last year that might be relevant to give you the context. I won't say too much about how these things work. How do I advance the slide? The arrow doesn't work. Oh, that arrow works, yes. I see. I think all you really need to know is that we generate X-rays as we do at a synchrotron by wiggling the electron beam, accelerated charges radiate. And when the wiggle period is equal to the X-ray period, you get a special resonance and you get gain. And this gain can be very large. It increases the intensity of the X-rays by perhaps 10 orders of magnitude. But these are pulses, and about 120 per second at the LCLS. Here's a picture most of you will have seen before showing the okay, in the middle. Okay, this doesn't work.
Yeah, it's this button.
Thank you. Showing the liquid delivery system with particles coming out of our nozzle sprayed across the pulsed X-ray beam, and in time resolved work we pump the particles first if they're sensitive to light before taking their snapshot. So the particles are running down here, the pulsed X-ray beam, hard X-ray beam running along this way. If there was a Bragg beam generated in the little crystallite here, it might run off in that direction, and then the highest angle would be limited by the angle of the cone. And here's an old picture from Uwe Barstow showing this liquid stream breaking up into droplets. This is an early pattern where by chance, the crystals of course are in random orientations. A crystal of photosystem 2 from Petra Fromm's lab happened to be lined up by chance along the C-axis. So we got this nice symmetrical diffraction pattern going out to about 3 angstroms. And that was back in 2011. It was the first indication that we could get high resolution data by this method. Now, there are over 100 structures in the PDB from XFEL results. I think about half of them have come from collaboration with BioXFEL. And there are 2 machines in the world. So as people say that at any moment in time on planet Earth, only 2 people can be doing an experiment at an XFEL, one in Japan and one in Stanford at SAC. So in a few years of work, there are more than 100 structures. Some of them are multiple depositions of the same structure, and about half of them have involved collaboration with BioXFELs. So just by way of quick examples, I thought I'd give you 3. This is work between a collaboration between Uwe Barstow and based mainly in Vadim Cherezov's lab. He's now at USC, and it involves angiotensin. This is a GPCR. It spans the membrane, and the cell is shown here by these dotted lines, and it signals to, on the interior, angiotensin 2, which has a role in controlling blood pressure. So this is all about drug delivery and ligand binding. You can see the small drug molecule is shown inset here. It's the stick figure here in yellow. And the binding of angiotensin 2 to this GPCR receptor, that's the AT1R shown here, is suppressed by the antagonist drug, ARB-ZD7155. This is an attempt to make an improvement on losartan, which is an existing commercially available drug for control of high blood pressure. So the ligand is also acts as an arrest and biased agonist, and which terminates the cycle of internal signaling. The internal cell is down here. Well, what was remarkable here was that getting good resolution. They managed to get 2.9 angstrom resolution versus 4 angstrom previously on a synchrotron, mainly due to twinning problems. And from this, they're able to make a density map. And of course, the pharmacologists are very interested in that because it helps them work towards drugs with fewer side effects. So that's an example of drug applications of an XFEL. There are others. And I've summarized them here. There are actually about 4 others. This is serotonin, which has been worked on in binding to an ergotamine ligand. Here's the smoothened receptor with the ligand of cyclopamine. And over here is membrane kinase. But in addition to that, the most recent paper has been on a delta opiate, which is one of the new painkillers, of course, that are causing so much trouble with addiction. And this is an attempt to tackle that problem. And we have a paper, which I really don't have much time for, which in fact got us the most publicity in the last year, from Eric Xu, on human rhodopsin, the sensor of the pigment at the back of your eye responding to light. And we have other work by that on with Marius, which I'll mention. So he's having a lot of trouble with the buttons. Here is the work with Eric Xu. He's at the Van Andel Institute. This was published in Nature last year. And it shows human rhodopsin bound to mouse arrestin. So here's the arrestin is shown here. This is the rhodopsin spanning the cell. And the process is indicated roughly up here. So it's activated by light. This is the way your eye responds to light. And arrestin binds on the inside. There's some signaling, and then that is released. And he was able to get images, good images from the LCLS. This is not time-dependent work, but he could get good images of the bound state of this and compare that with previously published unbound structures, and so say something about the binding mechanism. Okay, let's move on to time-resolved work, and I'll show work that's about a year old now from Marius Schmidt's group. And he says this is the fastest camera in the world. And here's his wonderful photo of that process. Of course, the pulse duration of the LCLS is about, we normally work with about 40 femtoseconds. And if you work that out, it's a camera shutter. It's equivalent to having a camera shutter of 25 trillion snapshots per second. So these are very fast movies in that sense. In fact, we use the fast pulses mainly because we want to avoid radiation damage by outrunning damage, as you probably have heard. That's a more important characteristic than the time resolution in most cases. So it's the ability to avoid damage and therefore look at reactions at room temperature, which is important here. So the technology for that was developed a few years ago. It's shown here. We use a light beam to excite molecules traveling along a liquid jet here. After excitation, we take their X-ray snapshot further downstream at this point. And that's using the sample delivery system I showed earlier. And here's the pump laser, so this is called pump probe. And it's been a very complicated system to set up, and there have now been many systems, papers published on this, which Marius has been a leader. So to see that working, here is the pump laser exciting molecules as they come out of the nozzle in the liquid jet. This is a little crystal of ice that's formed growing down here, and here are the molecules being blown up and exploded by the X-ray beam, sending off their Bragg beams to the detector. This is work from Petra Fromm's group in Photosystem 1, early work, and she now has results on Photosystem 2. And the authors on that, Chris Cupid is here, and Chelsea and others. So this is beautiful work, which has been slowly getting better over the years, a big effort starting back in 2009. That's the undocking of ferredoxin and work back in 2011. Here's Marius's image, his movies published over a year ago. And in this case, it's photoactive yellow protein, which is famous for making good crystals out at 1.4 angstrom resolution. That's the reason this was chosen. And he has actually managed to collect what you might call a movie, a molecular movie. Again, the fast pulses give us the opportunities to get damage-free data at room temperature. So the importance of that is you have the right value of KT applied to this molecular machine that it would have under physiological conditions, or even if it's a leaf in sunlight. But I think the important point, before I start this and show it to you, just let me say the colors over here correspond to the colors in the diagram here, which shows the reaction path. It's a cycle. But what we're imaging here is really chemical kinetics. These are images of kinetics. That is to say, the total amount of species as a function of time. We don't actually see the molecule jumping from one structure to another. That happens if you think about the rate equations, molecular dynamics. That happens very rarely and is extremely fast, this femtosecond transition. So what we see here are the amounts of the different species, and there will be several as a function of time. But we see this superimposed, the sum of those, in a spatial periodic average. Now, if you want to make a true molecular movie and see how the atoms jump around when a molecule changes from one molecule to another, you would need to do it by modeling. But we have important information here to base that modeling on because it shows us, as it were, the initial and final state in the kinetics between these stable intermediate phases. So the stable intermediates are labeled here, these symbols like PR1, ICT, PB1, and so forth. They're colored here, and the same colors are used here. So if I start the movie, you'll see the colors change. Remember, this is a spatial periodic average of the molecules in the crystal. There'll be three kinds of molecules: those that aren't affected by the pump light, those that are excited and set off on this chemical pathway, and those that are this chemical pathway, and a third type that follow the other chemical trajectory. Some, but the quantum efficiency is not 100%, so some do nothing at all. So we're now in this purpley, mauvey, bluey color here, which is ICT, corresponds to this one. The time interval is indicated in the bottom left corner. And as time goes by, we'll move slowly around this loop up back to the initial state. You see this purple color here, which corresponds to the PR1. And so it continues around to the beginning, and then the cycle continues. Now, in photosynthesis, there's another similar cycle. It's called the Koch cycle, where similar things happen. Those crystals are much harder to get high resolution from, but there's analogous work going on in Petra From's group for photosystem 1 and 2. So this is the best we've been able to do towards a molecular movie, and I think it's absolutely remarkable. But again, the thing to keep in mind is that this can be done at room temperature without damage. There are many previous studies where people quench in particular states, and you have to ask whether the quenching works and so forth. But the key unique capability we have here is to separate out any damage effects by minimizing them, by avoiding them, from the effects of temperature, which is driving the reaction. So see the reaction as it actually occurs in nature. And by modeling, Maris's group, by a heroic feat of modeling, has managed to fit the various species to the density map as a function of time. He uses single value decomposition, and he gets, as you can imagine, with rate equations, exponential growth. For example, this species PR2, and then it levels off and decays. Another species is generated here and dies out, and so forth. And so you can get the concentrations as a function of the stable intermediates as a function of time, which is a fabulous achievement. Now, it has to be said that there was earlier work at Biocar's using the Lowry method, but there you don't have this advantage of outrunning radiation. For many biological processes, they're not light sensitive unless you make them so, and so you really have much longer times to deal with, the microsecond, millisecond time span. For example, I think in an AIDS infection, it takes a full second for the virus to inject its genome into the cell. So the times can be very long, and you have plenty of time to do mixing of species. If we think of a typical experiment where we mix a substrate with an enzyme here, and the enzyme could be in the form of crystals, we can make a device like this, which was the Ph.D. work of my student. Much better versions have been made since in Noah's Pollock's group, in Cornell, in DAISY. And they haven't been published yet, but they're starting to work, and you'll hear results from that later. But the idea of this, there are two new things about this. Previously, people had worked on synchrotrons with solutions in cells, so that's mixing cell work, right? Now, when you do X-ray diffraction of synchrotron from a solution, you tend to, it works, but it's called SAX, and this will be fast, time-resolved, pump probe SAX. But it's poor resolution because you have to deal with the rotational averaging of all the species in the solution. Our idea, the question we asked ourselves back in wherever it was about 5 years ago, is could you do this with crystals? Because then you can get atomic resolution. So again, we have two crucial things here. We can get atomic resolution by using nanocrystals instead of solutions, fast SACs, and second, we can outrun the radiation damage. But a third important point, and Marius has written a paper about this, is that the diffusion of the species into the little crystals, if the crystals are small enough, is fast enough not to limit the time resolution. And that's a critical point. So we can use diffusive mixing and work at physiological temperatures and avoid radiation damage and get atomic resolution. That's the point. That's what's new about this work. And we have our first results. And I think I've got the wrong picture there, but you'll have to forgive me. And this is how this thing was meant to work. If you wanted a different species would mix here between, say, an enzyme and substrate. They'd react over a certain distance here as they flow downstream and come out here, turn into little droplets, and we'd take their snapshot. But it was a bit too simple. We needed to think more carefully about this because there's a dilution effect which has to be taken care of, and a more sophisticated design is needed. But this thing did sort of work. We tested with a fluorescent dye and a quenching medium, and you could see how far the dye would flow before it was quenched to measure this diffusion time, and therefore the speed, the time resolution of the device. But this is the wrong picture to show the first test of something like this at a very intense beam time we had last year, where we tried out all these gadgets. At the last minute, as always happened, we got something to work. And using Marius's sample of beta-lactamase, it was possible to see this drug, CEF, fully bound. Now, this is the last stage in the reaction after the full chemical reaction has taken place. Bound somewhere in the middle here, it's pulled out and shown enlarged over here. And the paper for that has been written, and this work is unpublished. So this probably should not be on the web. That's a comment to Tom, right? You need to remove this. Thanks. We certainly don't want this on the web because it's the wrong picture anyway, and it hasn't been published. Two reasons why it must not be shown. Let me move on. The last topic then is to report on progress on what's called the single particle initiative. And that's been going on for some time. The sort of poster child experiment of the XFEL back when it was first built, when plans were first made to build it around 2004, was that it would give us X-ray diffraction patterns from individual particles, with one particle per shot. That could be, let's say, a virus. How far have we got with that? Well, here's a virus, and here's its diffraction pattern. And this was worked with Ilma-Schlichting a few years ago, and it's pretty good. You see lots of rings. The resolution is approximately equal to the size of the virus divided by the number of rings. And I think I counted 16 rings here. So that would mean we had about 120 angstrom resolution. And if you divide the size of the virus by 120 angstroms, you get the number of pixels on a side. Now this, of course, is far worse than is possible with cryo-EM. But Mike Dunn, in his wisdom, set aside special beam times which are unrefereed and where people from anywhere in the world can come over the last year and try to improve matters. That's called the Single Particle Initiative, and it's been dramatically successful in cleaning up the beamline, getting rid of background, and giving us high resolution data. You could compare this with solution scattering, and ask the following question: what is the best number of particles per shot? If we have a lot, it's called SACs, and we know the resolution is poor. If you have one, you can't do better than have the beam as small as the particle, because then every X-ray has a chance of being scattered by the particle. But on the other hand, it's hard to hit one particle, like shooting with a bullet at a particle a long way away. The tunnel was 3 miles long. That's a simplification, exaggeration, of course. But so the hit rate goes down, and typical hit rates in the solution, in the liquid jets, can be up to 40%. What's the hit rate in the LCP? Is it sort of 20, 30, something like that, right? When we do these experiments, it's a completely different world. You stand there in the control room, looking at the screen, and nothing happens. Then every now and then a cheer goes up as you get one pattern. So the hit rate is 0.1%. So we're going to have a workshop on this crucial problem of sample delivery, improving the hit rate in single particle work, as part of our conference in Las Vegas in January. But there's been some very promising advances there. notably by Rick Kirian and the group in Uppsala, Jonas Hoydu, who are using electrospray and focusing jets and all kinds of clever ideas. You can run particles along a hollow beam of light, for example, kind of like a bit like laser tweezers. And there's some exciting developments there. So if we could, what we know is if we could get the hit rate up to even 10%, this would have succeeded. We would be competitive with cryo-EM. So it's all about the sample delivery, and that's what we're going to talk about in Las Vegas in January, in a one-day workshop. Okay, this particle was made by, I think, Brenda Ho, perhaps, at ASU. But you can reconstruct those diffraction patterns, and here's the best reconstruction I'm aware of to date, although very recently Absalma's group's done very well. This was done by Andrew Morgan in Melbourne, and it's taken from soft X-ray data, not the previous, like the previous. I'm sorry, yeah. This is soft X-ray, 1.6 kV in the MO chamber with a 2 micron beam, and it gives about 150 angstrom resolution. So what was exciting to us about this 3D reconstruction? You have to imagine how difficult this is. There's one particle per shot. You can't merge the data until you know the orientation relationship between successive shots. And errors in that appear as a loss of resolution. Now, very smart people have thought about that problem, and there are two main classes of algorithms, and on simulated data they work perfectly, and on experimental work data they don't work very well at all. So you have this problem, the same problem arises in cryo-EM. Our problem is different. We have Friedel symmetry in our diffraction patterns. They don't have Friedel symmetry in their real space images that they work with. And we have a phase problem, which they don't have, because imaging solves the phase problem. So it's more difficult, but there are methods to solve those problems. In cryo-EM, a critical problem is how to distinguish a change in orientation of a particle lying in an ice film from a change in conformation. And that's crucial because the algorithm has to decide if it sees two particles that are slightly different, are they the same particle in different orientation or different particles in the same orientation? And according to the errors in that, so you will lose resolution. Now the key to that is understanding that the internal structure of a protein doesn't change when it changes orientation, but it does change when it changes conformation. So you have something that's different in the two cases to work on. This is very tricky stuff, and they're very sophisticated algorithms. And believe me, all that's been learned in cryo-EM is being applied to the algorithms on the single particle XFEL project, particularly by the Hoydu group in Uppsala. So this is the best that's been done, and it was important because it's got knobs on it. Previously, all our reconstructions had been, blobology, and it was said of cryo-EM until a few years ago, it's a waste of time, it's all blobology. You always reconstruct things that look like blobs, and they have no new biology or information that's worthwhile. Well, for the first time, we have the icosahedral symmetry here in this picture coming out of the data. Because of cleaning up the beamline under the single particle initiative, a crucial thing was Henry Chapman's idea of masking apertures to get rid of the background. Also characterization, much more accurate characterization of the detector, and choose the choice of a good sample, which it makes where all the particles are pretty much identical. And that was a good search over many groups around the world to find the best candidate test particle. Turned out to be this rice dwarf virus thing, where it just happens that the particles are all the degree of dehydration is the same, so the particles look the same size in their X-ray, as judged by their X-ray diffraction pattern. So that work continues, and there'll be a lot of publications, because the rules of the game are that Mike Dunn set up were that it's an open collaboration. BioXFEL is a large part of it. Delano Salvin has done very important work on that, and been to the beam times. We go to the beam times, we get the data, and it's then published in this new publishing racket called Nature Data, Science Data, right? They got a new market, publish everybody's data, open source, so you pay the $5,000 to publish it. Author pays, right? So that's happening now, and then there's a race for every group to reconstruct the data and publish their results, and that's starting now. So we will see over the next 6 months what the best theoreticians around the world can do with this kind of data. But for hard X-rays, there's been significant progress too. That was all soft X-rays. Back at the CXI hard X-ray chamber, the data now goes out to, this is wrong, it should be 5 angstroms. There's an error in this PowerPoint. And again, this is unpublished, so Tom, please remove this. But we realize there's a constant fight between the SACS people and the crystallographers about the definition of resolution. And the wrong one was used here. It should be 5 angstroms. Well, that's a huge advance on what we were getting about 3 years ago. Now, we couldn't get enough patterns at 3 angstrom resolution to make a 3-dimensional reconstruction. Now, these patterns have been merged together. So you could ask the following question: how did you merge them without knowing their orientation? Well, the answer is the orientation was not determined. And to measure resolution, you'd have to merge them in their correct relative orientations. That was not done. But there's so few counts out here at 5 angstroms, you don't have to worry about it. So this is not really resolution. It's not strictly correct to call this resolution. We can say there's signal at 5 angstroms after merging, I think, 124 patterns without regard to orientation. Okay, so that's a progress report. That's where we are. Okay, I'm nearly done now. So let me just say that in the international context, there are machines being made around the world, and here they're all listed. The next ones will come on fairly soon in Korea, South Korea, in Switzerland, and of course the machine in Hamburg. All start up next year. And the one in Switzerland is rather like the American one. And there are other plans for other machines. There's a soft X-ray machine in Hamburg, and they are the main ones that everyone will be able to use in our BioXFEL. We expect people to go to these machines from the United States in the BioXFEL program over the next few years. There's plans for a second American machine called LCS2 that will give you either 2 soft X-ray beams simultaneously or 1 hard and 1 soft. Now, and the new soft line is superconducting, so it has much higher repetition rate. So you can get high rep rate soft plus 120, the same as we already have, low rep rate hard. However, the enterprising people at SLAC have put up a proposal to BSAC and DOE to increase the X-ray energy of the superconducting line and make it hard X-rays. And that has a pretty good chance, I think, of getting through all the relative committees in Washington and Pat Damer and so forth. So I wouldn't be surprised if when the LCLS2 starts, it will also offer hard X-rays. So we'll have 2 hard X-ray lines then, one of them high repetition rate. Okay, I'm going to finish by telling people why you would use an XFEL, because the obvious question to ask is, why do I need an XFEL rather than a synchrotron? Well, I've hinted at the reasons throughout my talk. To avoid radiation damage, outrun damage, to look at room temperature structures and avoid the effects of freezing, which we call cooling examples, to get in cases where you do need better time resolution. And in remarkable, very recent work, Irish Smith's group has actually got a movie of the cis-trans isomerization process in photoactive yellow protein. It's the same process as the first event in human vision, but he's able to do that at 200 femtosecond time resolution, something which could not be done on a synchrotron. You can go to about 1,000 times better time resolution. So there are problems even in biology where you need a femtosecond time resolution to understand mechanisms at the atomic scale. Irreversible reactions can be studied. Crystals large enough. The problems that Ed talked about, cases where we only have little nanocrystals. I mentioned the optical pump laser absorption length is comparable with the crystal size, so we can do diffusive mixing. If you think about putting cream in your coffee, right? You would sit there forever to wait for the cream to mix throughout the coffee. That's diffusive mixing. Or a drop of ink in a beaker of water. It just goes in and sits there, doesn't it? That's Einstein 1905, diffusion. If you stir it, everyone stirs their coffee because you're impatient, you don't want to wait. That's the time difference between diffusive mixing and turbulent mixing. And because our nanocrystals are so small, we get away with diffusive mixing. Very important point. And finally, in some cases, this is still a bit anecdotal, but certainly if you talk to Alex Brunger, you'll find he believes that the resolution is better at an XFEL. Now, the reasons for that are very complicated. They'll depend on the particular protein, its defect structure, the dose you use, the diameter of the beam, the bimolar factor, and other things. So it's a complex problem. Here is the BioXFEL Research Consortium. About half the professors are biologists or structural biologists, and half of them are methods development people at different campuses around the US. I don't think we need to spend time on that. And let me end with this summary shown here.